Hi, Shannon over here. This is my presentation on CRISPR. So the students will learn terminology of biotechnology, the history of CRISPR, where it came from, how it's used, how the CRISPR-Cas9 system functions for gene editing, and how CRISPR is currently being used to manipulate genes and some hope for the future. So as an introduction activity to kind of get the students involved in thinking in terms of genome, they're going to have a 2-minute discussion with a partner, 1 minute per partner, and students need to read the following paragraph. So imagine you've been offered a deal from a genomics company. You can get a free genome sequence and analysis of all of your DNA that includes a report of your ancestry, traits, and medical profile. The medical profile tells you about diseases for which you have a low risk of getting and also those which you have a high risk of getting. Are you interested? Why or why not? Okay, so students need to discuss with their partner, 1 minute each partner. Do they want it? Why or why not? Okay, moving on. So some key terms. Genetic engineering is the process of manipulating genes and genomes. And then we have biotechnology, which is a field you could even study in college. And this is a process of manipulating organisms or their components for the purpose of making useful products. So this could be in animals and plants for crops and lots of different things we can do here. Bacteria, useful bacteria. Some tools that are used in genetic engineering that we need to know about are restriction enzymes. These are restriction endonucleases that are used to cut strands of DNA at specific locations called the restriction sites. And then restriction enzymes create what we call restriction fragments, and they have at least one sticky end or a single-stranded end that is available for bonding. And then we also have DNA ligase, and that's what joins these DNA fragments together. So go ahead and click on the link for the YouTube video and you can watch the YouTube video. I'm not going to play it during this, but you can watch it anytime. And that video just kind of gives you a brief introduction as we move into lots of big new words. So where did CRISPR come from? So CRISPR is clustered regularly interspaced short palindromic repeats. It is repetitive DNA sequences that they found in over 40% of bacteria and bacteria and archaea. CRISPR, together with the CRISPR-associated proteins called Cas, is part of the prokaryote's defense system against viruses. So just like we have an immune system, so do the prokaryotes. So it protects cells by allowing DNA sequences that are identical to past pathogens into their genome so it can recognize them if they invade. And we'll kind of go through a picture on the next slide. RNA called CRISPR RNA or cRNA is transcribed from the spacer sequence at the CRISPR locus or location. The cRNA directs the Cas proteins in the bacterial archaea to identify the foreign invading sequences and cleave it, killing the foreign invader. So this is a mechanism of adaptive immunity in bacteria and archaea. So it evolved to adapt and defend against foreign genetic material like the phages. So several examples of CRISPR pathways have been discovered in bacteria and archaea, but currently the type we use in our genome editing is mostly type 2 CRISPR-Cas9, which creates a double-strand break in the targeted DNA. Okay, so this picture shows stage 1, which is the acquisition or bringing in this foreign DNA. So you can see the red going in red, and there's our repeats with the foreign DNA in the middle. And then stage 2 is where we process and create this CRISPR RNA. So then this CRISPR RNA is guided to the invading virus where it can go ahead and cut the DNA on the invading virus or RNA. So what exactly is CRISPR in terms of the way we use it? So CRISPR is a gene editing technique that targets specific sections of DNA to make precise cuts and breaks at the target site that we've programmed it to go to. And it can make genes non-functional or even replace one version of a gene with another version that we want. Okay, sorry about that. So what is CRISPR? We just did this slide. So how does CRISPR work? So there's two main components. So the first component is a targeting system that finds the right place in the genome to cut. And this is achieved by a molecule called gRNA guide RNA, which has the same genetic sequence as the target genomic site. The second one is a component that makes the actual cut in the DNA. This consists of a DNA cutting enzyme called nuclease. In this specific case that we've been using it in gene editing, we use the Cas9 protein. When both of these components are delivered into the cell, the gRNA will bind to the target genomic site through complementary base pairing, meaning A's will bind with T's and G's will bind with C's. In the process, the gRNA helps bring in Cas9 to the target site to make the cut in the DNA double helix. The cell's natural DNA repair mechanism will close this gap, but because the process is not perfect, a few DNA bases will be added or deleted, and this ends up rendering the original gene, a gene variant linked to cancer or one that would be related to HIV, non-functional. So we want to turn those genes off. CRISPR can also be used to replace an undesired version of the gene, like the gene that caused the disease, with a desired copy. In this case, the desired version of the target gene can be placed into the cell along with gRNA and Cas9. The cell will then use this alternate sequence as a template to repair the broken DNA through the process of homologous recombination, copying the new sequence into the genome. So I'm going to show you the two pathways that naturally occur for gene repair in the cells on this slide. So the two possible pathways are non-homologous end joining on the left, which creates the knockouts. Okay, this is a little more error prone, and then it renders the genes not working. And then the homologous recombination shown on the right, we can create knock-in. So we can add in the template gene that we want into the sequence. So it's inefficient, but a little more accurate in terms of what we're replacing in the cut area. Okay, so why do we care about CRISPR? What's all this fuss about? So CRISPR is super important because CRISPR-Cas9 can be programmed to perform gene editing in mammalian cells. So it can edit our genes. So this is a real possibility for gene editing. And it's simpler and easier to use than other genome editing techniques. It's efficient, much more efficient, and happens a lot faster. So we can change a short RNA sequence into a target, into any site we want to edit in the genome. So it basically brings back gene therapy. Okay, so what is gene therapy in the first place? So gene therapy is a genetic technology that directly used to directly treat genetic causes of disease. So it's kind of been an aspiration for physicians, scientists, and patients for a long time. So some diseases like cystic fibrosis and sickle cell anemia are relatively well understood and caused by variants in single genes. So if the disease-causing gene can be corrected or replaced, we would hope we could cure the disease or at least prevent the disease from worsening. Gene therapy has been attempted since the 1990s. Less than a handful of these treatments have so far been approved by safety organizations. All of the approved treatments work by adding one or more working copies of a gene to a person whose own copies of the gene are not functioning properly. On the other hand, gene therapy based on genome editing techniques such as CRISPR makes it possible to directly alter an individual's original copy of the gene. Genome editing-based gene therapy can be used in other ways too, such as adding a new gene to a specific spot in the genome. So with cystic fibrosis, it's a genetic disorder where thick, sticky mucus in the respiratory pathways leads to problems with breathing, developmental delays, and infections. Cystic fibrosis is caused by a mutation in one gene called the CFTR. Treatments have been very hard to find, and it's been an anticipated target for gene therapy for a long time. So one of the main challenges with gene therapy is the ability to safely and effectively make the desired genetic changes to all the cells that need the gene to function. In the case of cystic fibrosis, all of the cells in the lungs and sweat glands may need functional CFTR in order to properly produce mucus and secretions. While no therapies have yet been approved for use in humans, we have had some promising experimental results in mice and pigs where the majority of the symptoms of the disease have actually been reversed. Okay, with sickle cell anemia seen on the left side, this is a genetic condition characterized by mutations in the oxygen-carrying proteins in the red blood cells called hemoglobins. SCD causes hemoglobin proteins to stick together and lead to the red blood cells turning into a sickle cell shape. The sickle cell disease occurs because of a mutation in the adult hemoglobin gene, which is responsible for making hemoglobin from the time we are babies onward through adulthood. Gene therapy may be possible for SCD by directly repairing the mutation in this gene. There's also another promising approach which we could take advantage of, a second hemoglobin gene that functions in fetuses and then gets turned off shortly after we're born. This hemoglobin gene, called the fetal hemoglobin gene, is not affected by mutations that cause SCD. So one potential gene therapy to treat SCD would be turning on the fetal hemoglobin gene and turning off the adult version. Experiments using this approach seem to work well in mice, and clinical trials in humans should begin soon. So that's exciting. Okay. So the next question, should scientists alter mosquito genes? So malaria has caused over 445,000 deaths in 2016 alone. Scientists have been able to use CRISPR to cut a gene that allows the parasite to survive in mosquitoes. So cutting the gene also delayed growth and development, so we won't be able to survive in the wild. So that's not potentially the solution right now. But is this something you think we should do, is alter these mosquito genes? Okay. So moving on, researchers, this is super cool, have used genome editing to cure a type of liver disease in adult mice. So this disease affects 1 in 100,000 people, and it's caused by a mutation in a single gene called the FAH, where livers are unable to break down specific amino acids, leading to the liver failure. So scientists injected the CRISPR system along with working copies of the gene into the veins of adult mice. And happily enough, they were able to increase liver function of 33% of the cells, and this was enough to restore the lost function of the liver. So even though it didn't get all of the cells, we were able to do enough to restore liver function in the mice. So this is a possible treatment moving forward. I kind of race on in interest of time. So in 2015, a research group used CRISPR to make genetic changes in non-viable human embryos. So this is crazy. So they were able to use these embryos, and this study was so controversial that lots of people wouldn't even publish it. So they used non-viable embryos that could not have been developed into fetuses, but because a genetic change is made on an early stage in an embryo, it could affect the cell in the future of the individual, including the germ cells. So this form of germline genetic modification, this has led to a discussion and debate worldwide about whether germline editing in humans is appropriate and whether or how society should proceed with research and possible application. So this was to work on hypertonic cardiomyopathy cases, but pretty cool stuff. Okay, so for our closure activity, now we have another scenario. So the first 100 volunteers the company is offering to correct several of the disease-related genes found by analysis. Imagine this were a very new procedure approved by the government for safety, but without a great deal of long-term study. Would you volunteer for this added service? Okay, so it will fix all of your genetic-related diseases, or at least a couple of them. Would you volunteer? Why or why not? Go ahead and discuss with your partner. And this is it for my presentation. Hope you enjoyed it. Bye-bye.